Hello, I'm Sophia,
And I'm Emily.
and we are members of the University of Michigan's Synthetic Biology team.
We are submitting this project to Gogec. We will discuss our proposed development of a genetic test that will help to inform antiplatelet pharmacotherapy. So what problem are we trying to solve with this project? For a bit of background, when patients experience a clotting event such as a stroke or a pulmonary embolism or some other clinical issue of that nature, a physician will prescribe them antiplatelet therapies. So the first option for these class of medications is a combination drug of clopidogrel and aspirin, and this is currently the standard of care because there is the most clinical data to support it. However, there are also two other options for antiplatelet therapies, and these are ticagrelor and heparin. So to kind of add into the biology that backs up our project, the gene called F2PRL3, which codes for the PAR4 protein, has some alternate variants that occur in some different populations. So this variant manifests in the 120th amino acid in the PAR4 protein. So typically, and in most of the population, this codes for an alanine amino acid, but in some populations, it codes for a threonine amino acid. And data has shown that in Caucasians, who are the population that typically, unfortunately, makes up the majority of clinical trials, the alanine variant of this allele is most common, whereas in African Americans, we see that the threonine allele is more common, with about 57% of African Americans having this variant. And it's important to note that this specific variant is a single nucleotide polymorphism, which means that one single nucleotide in this gene experiences the variation, and this specific type of mutation is relevant for the means that we will use to detect it. So in terms of, we want to know what the effects of this variant to this gene are. And this kind of manifests in two different ways. The first is that we see increased activation of the PAR4 protein, which is a receptor on platelets. So increased activation ends up increasing the likelihood of clotting and increasing the patient that has this variant's prevalence or likelihood to different clotting events like a stroke or a pulmonary embolism. Furthermore, it does make them also more resistant to certain antiplatelet therapies. There is only ex vivo data to support this, and you can read more about the data that supports this second conclusion in our paper. So these two kind of come together to form this really big problem where people with this threonine variant end up being not only more likely to experience clotting events, but also harder to treat them. And this kind of manifests, this is something that contributes to data showing that there's a 30 to 60% higher incidence of venous thromboembolism in African American patients versus those of European descent. And there is also data to support that this PAR4 variant allele largely does contribute to this disparity. So kind of going back to our three antiplatelet options, patients with this threonine allele have different, as we mentioned, responses to these different drugs. So firstly, to the standard of care, clopidogrel and aspirin, these patients with the threonine allele experience less effectiveness in response to those medications. So clearly this is a problem because if you're prescribing every patient these two drugs that are the standard of care, but they don't work for 57% of African Americans, that will cause a huge disparity in terms of health outcomes. However, the second two medications that are less commonly prescribed do remain effective with the threonine allele. So this kind of disparity in how different medications affect these type of patients can be really useful to inform treatment approaches for them.
Our vision for this project is to develop a point-of-care genetic test for this variant threonine allele, which can be used in settings such as a primary care office, so that physicians can understand whether their patient has this variant. This knowledge can ultimately help physicians prescribe appropriate antiplatelet therapies that will maximize effectiveness for the patient. Our goal is to have this point-of-care test advance equity within medical decision-making by ensuring patients of all backgrounds are given the opportunity to receive optimal care. Our first aim within this project is to develop our assay in vitro by working with DNA fragment samples designed with varying alleles of the F2RL3 gene. We will do this by using an SMP LAMP method, or loop-mediated isothermal amplification method, which is described in a recent paper that we reference in our written proposal. This details an alternative to PCR that is cheaper and doesn't require a thermocycler, which we believe would be a good alternative to PCR in a low-resource setting such as a doctor's office. Based on the recent 2022 paper by Hyman et al., we plan to adapt this technique for detection by fluorescence of the specific single nucleotide polymorphism associated with higher risk of thromboembolism and less response to the clopidogrel aspirin treatment regimen.
The second aim of our project aims to go to this next level of instead just using our assay to detect the PAR4 mutation in simple DNA fragments. We'd like to do so in human cell lines, which will better mimic the kinds of assays that would be run in a clinic on patient samples. And the first step in doing this is kind of going over this hurdle in which we don't have a way, or a good way at least, to purify DNA from human cell lines in such a low resource setting like a primary care office. So the second aim aims to identify a way that physicians can use to do this. So our approach to doing this in terms of extracting DNA from cell lines so that we can detect the PAR4 mutation in it is inspired by literature that we read. You can read more about it in our written report, but it essentially uses a simple two-step method in order to extract this DNA. So the first is that we plan to lyse our cells using a mild chemical-based method, and then we plan to extract our DNA using a simple anion exchange technique. Then our next aim, aim 3, aims to use this DNA purification methodology in combination with the assay that we developed in aim 1 in order to employ this assay in human cell lines and detect the PAR4 mutation in a clinically relevant way. We aim to do this using a 2-phase process with a variety of human cell lines. So we will take all of the human cell lines that we are able to accumulate and divide them into 2 cohorts, a training cohort and a testing cohort. We will start with the training cohort, in which we will sequence the cell lines to understand whether each of them has the PAR4 mutation or not. We then plan to conduct our assay exactly how it has been designed with the previously mentioned DNA purification methodology, and then as well as the actual SMP LAMP assay that we talk about in aim 1. We will then calculate the statistical significance of the fluorescence difference between samples and cell lines that have the mutation versus those that don't. And our goal is for these 2 fluorescence levels to be statistically significantly different. However, it's significantly possible that they won't be, just as a product of using this assay in a fairly different setting. So if this is the case, we plan to tweak our constructs in order to better be more accurate in terms of the PAR4 mutation detection. And then we plan to continually iterate this process after we tweak our constructs by reperforming our assay and then continuing to tweak and reperform as needed, and kind of going with this iterative engineering process in order to optimize the efficiency of our assay. Then, once we feel it's sufficiently optimized, we plan to calculate a fluorescence detection threshold that would be essentially between the fluorescence level of PAR4 variant negative samples and PAR4 variant positive samples. And this will be used in clinic by physicians. So if your patient, if you run your patient's test and they have a fluorescence level that is above this threshold, you know that they have this threonine allele, and if their fluorescence level is below this threshold, then you know that they do not have this threonine allele, and this can help you make prescription decisions appropriately. Then, once we have our assay optimized and we've identified this fluorescence threshold, we will move on to the second part of this, which is the testing cohort. So we will use a similar approach where we again sample or sequence all of our cell lines in order to understand whether or not they have the PAR4 mutation. And then we will conduct our optimized assay that we just did on the training cohort. However, we will do this blindly in such a way that kind of mimics how random patient samples might be run in a clinic. Then we will compare the data we get from our assay in comparison with the sequencing results in terms of the PAR4 mutation detection, and then evaluate our test accuracy by producing some percentage of our cell lines that produced the correct results. Next, we will be discussing human-centered design, which has been an integral part of our project proposal and also something that we appreciate that Gogec values so much. So beginning with our progress thus far throughout the planning process of this project in terms of our human-centered design efforts. So firstly, we feel that the entire foundational goal of this project is really based in human-centered design. So the entire idea, as we mentioned in the introduction, behind creating this point-of-care genetic test is that it will enable doctors to have greater prescription efficacy and ensure that they have equality when they're making medical decisions. We feel that this is in the best interest of not only the doctor to be the very best physician that they can be, but also in the interest of the patient to receive the best medical care possible. And both of these are really great examples of human-centered design. The next thing that we have already been accounting for and thinking about in terms of human-centered design is the cost and ease of use of our assay. So we understand that our test is likely going to be used by people who have little to no wet lab experience, such as a tech in a doctor's office. And so understanding that, firstly, in order for doctors to even be willing to purchase our assay, it needs to be relatively low cost. And then secondly, in order, once they have the assay in their doctor's office, in order to be able to implement it consistently and for it to be able to work quickly enough and easily enough that their staff will actually implement it into day-to-day treatment of patients, we need to make sure that it is easy to use. And these are two really key qualities that we've been considering as we've been designing our project. And then the last component that we have been really prioritizing as we've been coming up for the methodologies we will employ in our project is to make sure that we're prioritizing accuracy of our test. Clearly, using our test to inform prescription decisions is a pretty high stakes use case of our test, and one that if a patient is misdiagnosed with the PAR4 variant allele could have pretty dramatic effects on their treatment regimen. So we plan to really highly prioritize the accuracy of our test and make sure that that is our number one concern throughout its design and development. We will next talk about our planned initiatives in terms of human-centered design for when we actually do conduct this project. So the first is that we'd like to consult with primary care physicians that interact with these kinds of antiplatelet or clotting medications on a relatively frequent basis. So we'd like to understand understanding why clopidogrel and aspirin is the standard of care, understanding if this PAR4 mutation is already thought about in any way, understanding if they would even be willing to use our genetic test, and if there's anything we can do or think about during the development of our genetic test in order to make it more likely that physicians will adopt it in practice. Next, we plan to speak with platelet biology experts. We've already collaborated with a few different at the University of Michigan in order to develop the idea for this project. But we feel that their expertise in terms of platelet biology at large, but also this PAR4 mutation and its effect on clotting events and drug efficacy and whatnot, will really help to make sure that our project is the most accurate that it can be and ensure that its results will be able to be translated in the future. And lastly, we'd like to collaborate with the developers of the SNP LAMP protocol in order to optimize the efficiency of our assay. So because this SNP LAMP protocol is relatively new, only being published in 2022, we feel that the lab that developed it would be particularly interested in helping us develop our project in order to provide them with also additional data that this protocol does in fact work. So we feel that collaborating with them would not only really help them and be of interest to them and something that they'd be willing to do, but also that would really help us to optimize this project and make it the best that it can be. Here are our references, and thank you for listening to our presentation, and we're really excited to speak with you all at the Gogec conference. Thank you so much.